Interphase. In the S phase of interphase, each of the chromosomes in the homologous pairs replicate. Chromosomes are the structures of nucleic acids and proteins found in the nucleus of cells that carries genetic information in the form of genes. The replication of forms the identical and duplicated chromatids called sister chromatids. The sister chromatids are connected by a centromere. Homologous pairs are basically chromosome pairs, one from each parent, that are similar in length, gene position, and centromere location. The centromere is the part of a chromosome where the kinetochore is. The microtubules of a spindle fiber attach themselves to the kinetochore during cell division. We will discuss this more in depth in prophase 1. The cell becomes a diploid, which is basically just a cell with two copies of each chromosome.
The centrosomes start developing the spindle fibers and they start pushing apart, moving towards the opposite sides of the chromosomes. The homologous pairs start lining up with each other so that the homologous points of the chromosomes start the crossing over process and the tetras are formed, which are the two pairs of sister chromatids you see crossing over right now. The crossing over process is basically when the homologous pairs line up and genetic recombination happens. The homologous points of two chromosomes will cross over each other, and the DNA molecules of non-sister chromatids are broken by proteins and rejoined to each other. It's important to note that the crossovers are completely random. Also, the chiasmata is the X-shaped region where the crossovers occur. So as you can see, the nuclear envelope starts disappearing.
Now the centromeres are on opposite sides of the cell itself, and the spindle fibers attach themselves to the kinetochores. Kinetochores are just protein structures on the centromeres, and move the centromeres towards the metaphase plate. Each chromosome attaches to microtubules from just one pole of the spindle, and the two homologues of a pair bind to microtubules from opposite poles. Orientation at the metaphase plate is completely random. Phase 1, the homologues separate and the sister chromatids stay together. The spindle fibers guide the homologues to separate poles.
In telophase 1, the chromosomes have arrived at the separate poles so that each half of the cells has a complete set of duplicated chromosomes. In animal cells, like the one we depicted, a cleavage furrow forms, and in plant cells, a cell plate forms. Cytokinesis usually occurs at the same time as telophase 1. Basically, it's when the two haploid daughter cells form. Here, meiosis 2 starts, and it's important to remember that cells move to meiosis 2 without copying their DNA. These cells are haploid, which means they just have one chromosome from each homologous pair, but their chromosomes still consist of two sister chromatids. In meiosis 2, the sister chromatids separate, making haploid cells with non-duplicated chromosomes.
In prophase 2, the chromosomes condense, the centrosomes begin to move apart, and the spindle begins to form. The spindle microtubules start capturing chromosomes and moving them towards the metaphase plate. Now in metaphase 2, the chromosomes are lined up at the metaphase plate, and the kinetochores are attached to the microtubules extending from each pole, just like in metaphase 1. Now that the sister chromatids have crossed over in metaphase 1, they are very genetically different, resulting in the randomness during sexual reproduction.
Proteins break the centromeres down in anaphase 2, and the sister chromatids finally separate. The separated parts move towards each pole and become chromosomes.
In telophase 2, the chromosomes decondense and nuclear membranes form around them. Like the cytokinesis during telophase 1, new cells are formed, but this time it's 4 haploid cells and each chromosome is just one chromatid, so it's not duplicated anymore. A haploid is a cell with a single set of unpaired chromosomes. This is the final stage of meiosis, and the cell becomes a gamete, ready for sexual reproduction. Gametes are formally defined as matured haploid male or female germ cells that are able to unite with another of the opposite sex in sexual reproduction.